let's just suppose you knew nothing of museums. This is what I think museums are about. Okay, so the most important question in zoology is a lot of what this is about, and we'll see how you go anticipating what that question is. So museums, including ours, the one I'm talking about, and this is all taken from its website pages. Everybody understands, because they were kids, they went to the museum with the school group, what the museum galleries are about. The museum's a place where you go to find stuff out, and it's laid out there for you, and there are all kinds of ancillary structures designed to help you learn more about the natural world and cultures in the case of a museum like the South Australian Museum. But in fact, that's just part of its role, and it's kind of a relatively minor part in some ways, because it's the derivative role. It is able to do these things because of the background work that is going on in the rest of the iceberg, so to speak. And so they have and more natural history museums have become the sole point for this very fundamental basis for the rest of animal biology. So every kind of biology you can think of comes back to this most important question. And it's the museum's job to answer it. And for this incredible amount of animal diversity in the world, this most important question is this one. This is what the museum does for the rest of biology. All of the high-powered theoretical work that goes on in biology assumes at the very base that when it says it's working on the zebra danio fish, that that's actually what it is and not some close relative or some similar thing. And so the absolute centrality of species identification is at the heart of all of biology. It's the basic communication device. And more than any other single organisation, it's museums' responsibility, really, to document that. Either directly through its own staff or by having the material available where staff from other institutions can come and reliably work on that particular question. So the biological species then is the basic unit of biodiversity. Whenever we're talking about biodiversity, we're implicitly talking about species, which means we imply that we know what we're talking about when we're talking about species in a particular group. And it's not just a label, it's also a summary statement. Once you know what a genus and species is and how that sort of biological classification stuff works, it immediately tells you 47 different things about that animal, just knowing that it's one of this group and has these sort of things as relatives. So that it's a species, getting the species right is also immediately having at your fingertips a summary of a whole lot of aspects of the animal's biology, where it's known. Plenty of species that that's not truthful, but that's the aim. And I'm a vertebrate person, so I'm in a kind of a cushy position compared with a lot of entomologists, for instance, because by and large we know a lot more about your average vertebrate than we do about your average insect. So you'll have to kind of imagine that someday entomologists will be as well off for information as vertebrate biologists are. And in terms of the rest of biology, the theoretical basis of biology, how the animal kingdom works, where it comes from, why it is like it is, this is all based on species as well, ultimately. So, you know, these days, species is more than just what a taxonomist says it is. Now it's a demonstrable gene pool. That gene pool doesn't evolve with that neighbouring gene pool because they tend not to swap genes. And so each species then is its own little evolutionary experiment on its own journey through time. I do have a butterfly illustration. Here it is. I'm sorry, this is the only one. Things are going to get a lot scalier after this. But again, this is just to remind you that there is an actual task involved in winnowing out all that variation and trying to decide what's important and what's not, and using all the new tools that we're getting in genetics and other areas of biology, all the new micro tools and things like CT scanning and stuff like that that I've been using, that enable you to analyse and assess and sort of statistically establish what's in and what's out in terms of where the useful information is about where this group of species are stopping and starting. And so, for all of this stuff, in the end, you need some kind of ability to go and get some diversity quickly, to go and assess it, and if you had to go out into the field every single time to catch the creature that you wanted to look at, you'd have you need a very long lifetime in order to get that information. By storing specimens so that they can be repeatedly accessed over time, museums provide this instant ability to walk into what you could call nature's supermarket and just get stuff off the shelves so that you can immediately start to get the picture of what's going on. Oops, let me just do this a bit there. That's right, okay. And so the objects themselves are studied by people interested in diversity, but also anybody working in biology, establishing some sort of fact about some sort of animal, can always take the option of lodging one or two of that study animal in the museum at that time. And from then on, anybody who wonders which of this species complex did that guy study back in 1956, well, he put some specimens in and there it is, and it's this one and not that one, and so on. So you've got this capacity over time for quality control, in the absence of which many biological findings start to drift off into speculation as knowledge changes over time. But being able to sheet it home, and there has to be a place that everyone agrees you could go to and do these things and lodge this material and go and see it. And that place, it's been determined kind of by the community over the last century or two, is a natural history museum. So all the natural history museums of the world, to some extent, are a single entity whose job it is to preserve and document the world's animal diversity. It's a time capsule as well. So this is one of the animals I've been involved with over my time at the museum, the pygmy blue-tongued lizard. This is an animal that was described in the 1860s, but throughout the 20th century, up until 1992, there were no wild populations known. So up until that point, anybody who wanted to know anything about a pygmy blue-tongue, they had to go and look at one of the 20 or so specimens that existed in several different museums. Those 20 specimens gave people a couple of locations, so the original type locality area, so if you want to see animals from that Gawler area, you have to go to Berlin, where the original specimens are. A specimen found its way into the Vienna Museum from Seven Hills up near Clare, and then the South Australian Museum has a few here and there, a specimen from the Dry Creek Rail Station. A school teacher from Burra, when he retired, brought the contents of his nature table down, and there was a couple of pygmy blue-tongues there from somewhere around Burra. And the very last thing before 1992, some guys at a building site at Mitchell Park found a couple in what had been a yard of a small inner sort of to the city farm that was being turned into suburbs at the time. We now know that they're still there and they're up in the mid-north, but those southern populations no longer exist. Everything we know about them depends on that couple of specimens. And of course our knowledge keeps on changing over time. And so the example I've got here is a fairly familiar one. This is the bearded dragon. And again, from its original description up until later on in the 20th century, this was regarded as a single widespread lizard, very distinctive with its big beard and its threat display. And then a PhD student came along and she found that there was actually two species involved in this thing we'd always thought was just one. And it wasn't just someone splitting hairs. These things were different in quite a few significant ways. Pretty soon, other workers came along, changed the genus that it was in, and now those two species in South Australia, we know they have these sorts of distributions. One's a very inland, dry-adapted species. One's a much more temperate, sort of even subtropical species. And the two of them differ in lots of different ways. They have different looking males, different sized beards, different sorts of habitat preferences. One of them, it turns out, makes a brilliant pet. And now the world has inland bearded dragons for pets. There are magazines devoted to bearded dragons, just like there's magazines devoted to budgies and zebra finches and cockatiels, some of those other inland animals that Australia has given to the world as pets. Real Pagona barbata remains a difficult animal to breed in captivity. It doesn't settle down very well. It's kind of high-strung. The point is that the distinctions made by taxonomists actually feed into all sorts of aspects of the biology of an animal. This is just one example. Until you get the names right, you don't have those insights. Museums are often the only way we know the distributions of an organism. Because most people can't identify most creatures, it takes the experts to be able to identify these things, and they can use museum specimens to do this, to make distributional maps that improve over time as more specimens become available and so on. But the other thing that happens over time is the sort of thing that happened with the bearded dragons. People start to discover that what they thought was one widespread species turns out to be a complex of multiple species. And over the years since Ctenophorus maculatus, as we would call it today, was first described, it's been realised that some of these populations represent actually different species. And in fact, they represent about 11 different species. It didn't all happen overnight, but it did happen. And one of the problems then is, well, one of those things is Ctenophorus maculatus, which one is it? And this is again where museums have an essential, fundamental role to play, because every name, as it's established, is based on what's called a type specimen. So one particular specimen is put aside and that becomes the ultimate reference example for that species. And who holds those reference examples? Museums do. Museums hold them because a person lodging it with a museum understands that that museum is forever. It's going to be there with that job for the rest of research time as far as they're concerned. Museum departments, university departments and government departments come and go, and the material they acquire accumulates, then is thrown out, accumulates, then is thrown out. Museums are organised to look after collections permanently. So when it comes down to it then, the type specimen for Ctenophorus maculatus, which used to be Grammatophora maculata, is in the Paris Museum. It's in the Museum National d'Histoire Naturelle in Paris. And it was collected in 1818. And the collection locality is not too bad by the standards of some of these things. It says Shark Bay, which is a pretty exact chunk of the Australian coast. So that's a very encouraging thing to find. It's also encouraging to see the specimen still in pretty good condition. As it happens though, at Shark Bay there are two species. There's one that's quite limited to Shark Bay itself and the immediate area around Carnarvon in this sort of coastal complex in the yellow, and the more widespread sort of hinterland species, but nevertheless comes onto the coast and actually occupies the Peron Peninsula. So on the two Shark Bay peninsulas you've actually got representatives of two different species. Fortunately though, they're not actually, they're not completely the same. We know they're the same genetically, but they're not the same also physically. There's one paler species, but this is the one that lives in the yellow area. See, there's its yellow outline. It's me being organised. And this is the species in the blue area, something often more boldly marked, more heavy black markings on its back. That helps a bit, but what really helps, as it turns out, is male advertisement colouring. So the paler animal males have got a dark throat, with yellow trim around the edges. These boldly marked animals have a black chevron with a big black kind of hairy chest mark as well. And if we go back and look at that type specimen, that's the kind of markings on that specimen. So the blue guys are real maculatus. The other guy gets another name, he gets Tenophorus bardius, and that's how this stuff kind of works. And unless you have these type specimens, nomenclature will just fall into chaos. Now, taxonomic changes. I've been talking about various taxonomic changes, splitting things up into different groups as we get more and more precise information. That's the sort of activity that at times has been poo-pooed by other biologists or by other people. You know, crazy obsessives in their little garrets worrying away at exactly how many angels you can fit on the head of a pin, that sort of thing. But remembering that these names are keys to knowledge about the animals, it shouldn't be surprising that when you have a taxonomic change, it can have all kinds of other ramifications. And the first one I want to talk about here is conservation, that thing that you guys are talking about most weeks. So here's a thing I was involved with early in my career, being based in Melbourne and looking at temperate lizards. There's a widespread animal going from sea level up to alpine areas that is pretty much a map of the cool temperate zone in southern Australia. At the time I started working with it, it was a single species with what were regarded as two colour phases, one more broken up and speckly, one more continuous and more clear lined. As we started to work on these, and we were doing integrated studies, we had genetic information coming in as well as re-looking at the animal's exterior. We had chromosomal information, so we had quite an arsenal that we could throw at it. What we actually found, in fact, was that there were 5 species. 2 of them had speckled patterns, 3 of them had striped patterns. They weren't all identical. These 2 colour categories, it turns out, had an awful lot of slop within each one, and they were poorly defined, and so we were able to more precisely define the colour patterns, but also to show that there were chromosomal differences, there were genetic differences. These things were often occurring in up to 3 different species were overlapping in the same area in some cases. But from the conservation point of view, a single species with this distribution is not a conservation problem. This is a clearly, and also in detail when you had animals living in forests, in coastal complexes, in alpine meadows, you've got an animal that's a habitat generalist and is widespread. It's not a conservation problem. Once you have these 5 species, though, you can look at them individually, and what you find is, what we found was that several of them are very clearly conservation problems. So one I can mention here, the animal that became Pseudomoia rawlinsoni is a swamp specialist. This animal likes wet feet, and it's only found in remnant populations that represented by these little blobs. It's nowhere is it found throughout those blobs. It's just found within those blobs in little patches that are swampy or seasonally swampy. And most of the area where it occurs is now agricultural land, and most of that agricultural land doesn't tolerate swamps on their block. They've drained it and improved the pasture, and consequently glossy grass skinks no longer occur there. And so they occur in a handful of little sub-pockets within these little patches, and it's now agreed by all 3 states that have these animals that in each of those states it's more or less of conservation concern. And we only know that because we got the name right. That name brought with it all of this unknown biology which has now emerged, having got that name right. Bad taxonomy can kill. If you have not got these animals properly sorted, they'll go extinct without ever going on the record. Other aspects of getting the names right are a little more personal to us humans, if you like. So a name is a prediction, as I've said. A name is a prediction in this case of how bad would it be if one of these things bit me. And so common names, vernacular names, are kind of handy. but names like tiger snake or black snake might be hard to apply accurately, consistently. And just to not leave you in too much suspense, the top two animals are actually two different brown snake species.
Wow.
One of the bottom animals, that one, is a red-bellied black snake. That one's a tiger snake that happens to be all black. So, again, the deeper sort of taxonomic understanding of diversity and variation and how that goes is not just a matter of getting the label right. It's very satisfying to get the label right, of course, if you're one of the nerds like I am, but if you're one of the public faced by one of these things that's just bitten you, and you need to know that if you don't get the right antivenom, you might die, then, again, bad taxonomy can kill. So, keep on reminding yourselves and reminding anybody who asks that this business of getting the name right is the only way you'll have access to a whole lot of biological knowledge of these animals that might be crucial for the animal's survival, might even be crucial for your survival. So, implicitly, we've been talking about people going out and finding animals and discovering that they're new in various ways. But realistically, and as probably a lot of you know, a lot of the primary initial discovery doesn't happen out in the bush where a new thing suddenly appears under a rock or from behind a tree. It comes when people are looking at specimens that have already been collected and noticing anomalies of one sort or another, or because new information from one source stimulates a revisit to all of these collections. So I'll just take you through one rather recent study which has lots of different players in it in some ways, and just to show how, I suppose, species discovery happens, at least amongst the vertebrates these days. So this starts with a PhD student at the University of Michigan. But he's working with his professor who got his PhD doing fieldwork out in Australia on desert lizards, and so they're following up on desert lizard diversity in various ways. He's a theoretician in biology now, and so he uses some of these sort of examples to ask big questions about what are the broad processes that are going on that drive differentiation and evolution. Not really concerned with individual species much. The PhD student is getting some new gene sequence information where you get the entire genome, break it up into little, what are called SNPs, little single nucleotide polymorphisms, and you can look at the patterns of relationships across the whole genome from one species to another. And one of the species he's looking at is this very common desert animal called Tenotus schomburgii, and one of the specimens that he's looked at is rather divergent from all of the other ones. Doesn't necessarily mean anything, might just mean that it's the oldest one. I'll point out too, here's the specimen numbers for all these things he's looking at, and they're all from either the South Australian Museum or the Western Australian Museum. As I said, natural history museums are kind of like a global resource for biologists everywhere. And if you need a specimen, or in this case a tissue sample, that's been stored at a museum, very often it's fairly easy. What you have to do very often is simply ask, and you can receive that material. You can certainly receive specimens on exchange and so on. Anyway, so he's working on these. I get an email asking, is there anything odd about this particular specimen that might make us think that there was something going on? And so I was able to borrow that specimen from the Western Australian Museum over to the South Australian Museum, and there was a couple of things that one could see in that specimen. One is that it had more stripes. So normally there's 5 black dorsal stripes on Schomburgii. This one had 7. Still odd. If you look at their little faces, little pointy nose, fast-running desert lizards, the markings around the face are consistently different. So normal Schomburgii over here has a white stripe under its eye that runs right to the ear. This animal has a broken pattern on the side of its head. It has a black stripe above it such that all the area behind the eye is dark. this one has white that goes up and breaks it up. In fact, you end up with this sort of vertical series of spots along the cheeks as well very often. So it's consistently different looking in colour pattern, it's consistently different in genetics. Turns out, as I go through our collections, I can pull out other animals that don't have a lip stripe and have spots behind the eyes and have got 7 lines on the back, and there's 30 or 40 of them, all mixed up, all have come in as Schomburgii. Turns out that this particular appearance is consistent, and you can develop a map for where they occur. They're all from sand dune deserts in the Great Victoria Desert. And just to summarise, this single individual initially has put us onto the fact that there are 2 morphological groups, 2 different colour pattern groups. We have additional material genetically of some of these other specimens. They all fall out with that side branch as well. So they're genetically consistent, they're anatomically consistent. It turns out they're all from a particular kind of habitat. They're all from sand dunes, not just from randomly spread across the desert, which is, Schomburgii is a very widespread desert generalist. This thing is a sand dune specialist with its own colour pattern and its own genetic signal. It's a different species. And the reason we have that specimen in the first place is because of collaboration between 2 museums, the WA Museum and the SA Museum, because museums are constantly seeking to add to their material. They have gaps in their collection, gaps in diversity, gaps in geography. And museums are aiming to have everything in place should any question about any of these animals come up. So we were aware that the area in the far east of Western Australia up against the South Australian and Northern Territory border was a bit of a blank spot or an underappreciated area in terms of collections. So we organised to go there for a couple of weeks and simply add to what we knew from those areas. So lovely area, that area of the central ranges of eastern Western Australia. And we set up our camp. We do all the old style, African Queen kind of camping out in the scrub, and acquiring material. One of those acquisitions was that specimen that kicked off this whole taxonomic review. And again, just a reminder, Ivan Pratis from Brazil went to Michigan to study Australian lizards. And that's what's going on across the world now. It's possible because museums exist. If museums didn't exist, that didn't happen. And we'd still be none the wiser about how diverse our lizard fauna is in the deserts. And I'll just finish off with a last slide, really, just to sum up the sort of stuff that having a museum next door, like the University of Adelaide does, or like many overseas institutions where the big state museum turns out to be the big university museum, because that's the way it started, means that, again, the biodiversity supermarket is right next door, and if you've got queries and things that need answering based on what an animal looks like, you can go next door and do exactly that. So, for instance, Kate Sanders' group at the University of Adelaide are sea snake researchers. And a lot of their work means going out to Broome and places like that and doing various ecological things with sea snakes and putting them into pools and measuring things, measuring how they swim and looking at what they do. But they also do straight out taxonomic work and evolutionary relationship work, where you actually need to look at the bodies and count the scales and do all those sorts of things and get hold of specimens that you haven't got by borrowing them from the Netherlands or from New York or from London. And then in the museum, the specimens themselves with their scales can be examined closely. Jenna Crow-Riddell was a PhD student, and she started to look at the little sensory structures that are dotted all around the faces of sea snakes that look like little 5 o'clock shadow or paws all over their skin. And it's the snake equivalent of whiskers around the front of their face. She was able to discover, for instance, that sea snakes have light-sensitive tails. They have some of these sensory structures on their tails that are light-sensitive. Basically, warns them that their tail might be hanging out if they're hiding under a rock, and they can pull it in. At the museum, we have skull collections. What we've also had for the last 10 years are CT scans of skulls of representative species across the whole diversity. Alessandro Palci was visiting, was looking at some of these scans, and some of these skulls had this hole right in the middle of the brain case where normally it would be for the little pineal eye that is used as a light sensor in most lizards, which is interesting because snakes don't have one. So what's that hole that looks like a pineal eye but isn't? And what he found using our material was that, in fact, it's the outlet for a major blood vessel that takes a huge amount of circulation into the facial skin of sea snakes. So they have this vast network of blood vessels right below the surface, which is pretty ideally placed to be a little extra oxygen exchange surface. So they can hold their breath and exercise for a long time underwater. They can also top up their oxygen supplies to a certain extent, at least for the brain, by oxygen exchange directly diffusing into their circulation around their skin. We have some sea snakes have tiny heads. They're designed to forage into crevices and pull out fish that are hiding in there. The small-headed sea snakes, if you look at their skulls, you can see how they're related to the larger-headed sea snakes. And Emma Sherritt and her team have found that, in fact, small-headedness has evolved about 10 times. So people thought they were all a little related group of sea snakes. What they are is a repeated entry, where the rest of the skull's rather different, but they have in common the fact that the skull's very teeny tiny. So all of this stuff is based on the museum specimens. Without those specimens, some of the questions wouldn't have even been asked in the first place, and if they had been asked, they wouldn't have been able to be answered. So just summarising it again, in biology, a name is more than a convenience, it's a prediction. Knowledge is incomplete. It's going to need correcting into the future. We can't just stop now and say, that's good, we can just tell people about this from now on because we know enough. Not true. For knowledge to be knowledge, facts are best founded on something that can be checked, a specimen. Research collections are databases in 4 dimensions. They include time. The only way we're going to know very much about things like pig-footed bandicoots from now on is by looking at the specimens that we were lucky enough to acquire in collections before they became extinct in 1912 or thereabouts. The collection itself is a place of species discovery. It's not just a place where you store things, it's a place where you can go on a field trip into the stacks, into the drawers, into the jars. And properly managed collections of bodies, bones, tissues, images, they make possible broad areas of biological discovery. And so they're the only entities that exist to provide these services, and the role they play must be valued and supported. I hope you'll agree.